Let's go. So for this next module in the Leica SP5 Confocal, we're going to be demonstrating how to do a Zed Scan to get a three-dimensional representation of our image by using confocal light. So for our Zed Scan image on the left-hand panel here, we're going to look carefully at our acquisition mode to ensure that we've selected X, Y, Z. And that means we have potential 3 levels of scanning. And what you're going to see here is under normal situations, this bar will be collapsed, but we'll click on this tiny triangle. And we're now going to get a visual representation of the confocal volume that's about to be scanned. In order to achieve our Zed stack with this visual representation of the confocal volume, we're going to need to set our top and bottom levels. So our focal point at the top of our object and at the bottom of our object, and then we're going to scan in between those two objects to generate our Zed stack. And the way that's done is by using the live scan feature and adjusting the Z position back until we're just at the top of our image. I'm going to press stop, and we're going to press this arrow, which is going to then lock the top position. And what should happen is the arrow should turn red. We're then going to hit the live button again and focus our Z position through the object, through the middle of it, into the back of the object. Press stop, and once again click the arrow and it will turn red. So if your volume is properly set, you should see 2 red arrows. You should see your beginning point and your ending point, and they should be hopefully very different numbers. Then what's going to happen is the software is going to determine that, because we have this button selected for system optimization, the proper distance to stack in between the various levels given this fluorophore and given this objective, in order not to lose any light information between the stack. And what that says is that the optimal Z volume, which in this case we've selected at 3.2 microns, will have a step size of 0.13 micrometers for a total number of steps of 27. So if we do 27 steps within that volume, we will capture all the potential light information within that volume. In order to do that now, we press the start button, not the capture image button. We press start because we are now starting an experiment. And when I press start, you're going to see 2 things happen. You're going to see the repeated scanning of the image and every step down through the confocal volume, and you'll see a nice little progress bar appear here that shows us what step in the scan that we're at for the experiment. So as I press start, there's our progress bar, and here's our top scan. And what you should be seeing is with each progressive scan, you're going to get more and more in focus until you're going to get to the central focal point, which should be halfway through the stack. And then you're going to go out of focus again as you now focus on the back. Okay, so once the experiment's completed, you're going to see now there's multiple options to visualize the data. So when you're in Zed stack mode, and once a Zed stack has been completed, you're now going to see a slider appear here for a Z plane. And if we grab that slider, the red box, and slide it down, we can in fact manually scan through our Zed stack to the correct focal point in the middle, all the way to the bottom, and all the way to the top. Another way that can be visualized is by looking at all of the stacks along the series, which we can manually click on forwards and backwards. And a third way is by looking at X, Y, and Z dimensions, where we can now grab the crosshairs and look at the exact focal plane in XY by grabbing these hairs, or in Z. And so you can now focus in 3D space to the exact area of interest that you want to see, and you get an immediate visualization of what things look like in X and Y and in the Z planes, XZ and XY. In addition, this type of imaging can be used as a tool on very dim images, because what you can do is, instead of looking at it through the whole series of images, is you can generate what's called a maximum projection. And that's where all of the light information is compressed down to one focal plane from all of the images, and you can go from a very dim image to a very bright image, keeping in mind now you're looking through a much thicker confocal volume, and in fact you're more closely approaching what you would get from wide field imaging. And this is sort of a last resort option if you have a particularly dim sample. So by this methodology, you can do relatively straightforward Zed stacks. Combined with our other training module on multi-color imaging, you can do Zed stacks in multiple colors as well. That completes our module on XYZ imaging using the SP5 Confocal.